Thank you very much. And thanks so much for the National Organisation for Disorders of the Corpus Callosum for inviting me to speak. I have my colleague that I work very closely with here in the fourth row, Elliot Scher. We do genetic studies together. So I wanted to just introduce myself a little bit. I am a researcher a professor of neuroscience. I have been working with people with corpus callosum disorders for a long time in Australia. And then in 2021, I moved to the US to take up a position as a head of the Department of Neuroscience at Washington University in St. Louis. So one of the reasons I moved to WashU is that it is an amazing place to study neuroscience. It's really an incredible neuroscience community there. And I feel that there's a lot of interactions with clinicians and that I could really have even more of an impact in understanding corpus callosum disorders by moving to WashU. So we are doing studies in our lab at WashU and just getting things up and running there. But prior to moving here, of course, I've been working with Elliot for about 27 years on genetics of corpus callosum disorders. We've had a great partnership both in trying to discover the genes that are involved in corpus callosum disorders, but also my specialty is figuring out what the genes actually do in the brain. So if we could just have the first slide, please. Or you're going to display it like that. Okay. So today's talk, I was going to go over how corpus callosum disorders, how variable they are first, and then understanding a genetic diagnosis, some basics around that, and then how do genes cause disorders of the corpus callosum, give you a couple of examples, actually illustrating some work in animal models, so you get a flavour for how we actually figure out what genes do. Okay. All right. So here is a neurotypical individual. You can see the corpus callosum there. And here is a person with agenesis of the corpus callosum. Now, in Elliot's session just before, there's a bit of overlap in the session on hyperplasia where Elliot also had gone over some of the phenotypes. He talked about this structure, which is the lateral ventricles here, and this condition, which is called colpocephaly, where the back part, so this is the front, this is the back parts of the ventricles are enlarged, but the front parts are not. This is almost always occurring with complete agenesis of the corpus callosum, along with, of course, these bundles of fibers here. I don't want to stand in front of you guys. These bundles of fibers that you can see here that are known as Probst bundles. All right. Everybody is an expert in this room, I think so. Okay, so complete ACC and also partial ACC, of course, where the corpus callosum is small. But you can see every variation here from no fibers crossing the midline to a few fibers crossing the midline to almost a full corpus callosum. And then this condition back, which is a thin corpus callosum. I'm sorry, the contrast isn't very good there, but this is hyperplasia of the corpus callosum. This individual actually has partial and a little thin back part as well. So you could call this partial, or you could also call it hypergenesis of the corpus callosum. Okay. And so, as I mentioned, the typical features of these disorders are colpocephaly and the probe spundles. So where we see all three of those, we tend to call it, and complete ACC, we tend to call it isolated corpus callosum dysgenesis, meaning that there's these three are very typical. Doesn't mean there's only the corpus callosum, because you have the probe spundles and you have the corpuscephaly, but there's no other brain features that are changed here. And so I'll talk a little bit about corpus callosum disorders plus other brain disorders and how they arise a little bit later. I've got to point it at that. Okay, corpuscephaly. I think there's a bit of a delay. Okay, so in the research setting, we can do a different kind of scan. So I've been showing you these ones. These are called T1 weighted MRI scans. These colourful pictures that you can see here are from a different type of scan using the same kind of MRI scanner, but a different type of scan. So we use something called a diffusion scan. And essentially, in very my own simple terms, when somebody's lying in the scanner, we use pulses of radio waves, which help the water molecules within the tissue flip into alignment. And they flip into alignment preferentially along fiber tracks. And then we can use a computer program. So the data is like, they're called voxels. So we get this three-dimensional scan, and it's made up of things that you could think of like a pixel in a camera. And within each voxel, there'll be an orientation of the way the water molecules are diffusing. And then we use a computer program to map one voxel next to the next one, next to the next one. If they're lined up, it forms something called a streamline. So it's an artificial way of looking at the fiber tracks in the brain, but it's pretty accurate. So what we can do then is color code those fiber tracks based on their orientation. So if they're going across the midline, they're colored red here. If they're going from front to back, they're coloured green. And if they're going up and down, we colour them blue. That's a pseudo colour. That's a colour we put on the image, right? So here you can see the neurotypical, and we're looking at a slice this way. So we're looking down on top of the corpus callosum. So you can see the neurotypical here with the red fibres. And in this person with complete agenesis, you can see the fibres running now front to back instead of red. So they're coloured green. And the blue ones are the ones going up and down. And in this person, you can see partial agenesis. Now you can see these green fibres here. These are the probe spundles. But you can see a few little red fibres because this is partial, so there's a few fibres crossing. And this person actually, which we sometimes see in people with partial agenesis, is something called a sigmoid bundle. It's not normally seen in neurotypical individuals, but it's a connection from the frontal part of one hemisphere to the occipital part of the contralateral hemisphere. So we still don't know the function of these bundles, but I put it up there to illustrate the kind of plasticity that you see during brain development and in people with these brain wiring disorders. Oh. Okay, and again, the contrast is not great here, but this is a study from our group. We have the T1 images here, and here we're focusing on people with partial agenesis of the corpus callosum. So what we've done here, this might be a little bit easier. So there's 7 people here with partial agenesis, and we've just drawn a little schematic of just the midline part of their corpus callosum. So even from this, you can see these are 7 different people. You can see how variable the partial agenesis is. And then when we do this kind of tractography scan that I just showed you, we can then see which parts of the brain are actually connected via this corpus callosum remnant. And when we do that, that's these individuals over here. So these 3 panels are one person. This is the neurotypical. These 3 are from a different, ACC1, ACC2, ACC3, etc. And if you could see that image really well, and we will provide the slides, so maybe have a look again later if you're interested. But what this really shows is that when you do the tractography, so we can trace which areas are connected via this little callosal remnant, they're different in each person that we study. So what it means is that even if you had 5 people all with a callosal remnant that looked like that, they wouldn't necessarily have the same brain areas connected via that remnant. And this study first was done in Elliot Scher's lab. Lynn Paul's lab has also seen the same thing. We did this study on very high resolution MRI images. Well, that is great. Thank you so much for lowering the lights. We all see that it's highly variable. So we're very interested to understand how the connectivity, which is different among different people, actually relates to the cognitive outcome and that different people have. All right. So now, before I will get to the genetics, don't worry. But I just I'm still talking about how variable this is. So we can study the corpus callosum in mice. Now, mice are a really great model for corpus callosum development because A, they have a corpus callosum, and the early developmental events that form the corpus callosum are very highly evolutionarily conserved. And we have a lot of genetic technologies in mice such that we can take human mutations and make those same mutations in a mouse. And then we can study how does that mutation actually affect corpus callosum development. So this is a mouse, this one here, which is called BTBR. It's an inbred strain, and Elliot's group actually mapped the gene that causes corpus callosum dysgenesis in this mouse. It's a gene called Draxin. And this mouse has complete agenesis of the corpus callosum. Now, very interestingly, in mice, they tend to either have complete or full corpus callosum. And so we didn't have good model systems to study partial agenesis, but Elliot's group discovered that if he crossed this BTBR mouse with a wild type black mouse, and then you actually cross those pups back to a BTBR mouse. So it's a bit of genetic trickery here. But what you can get are litters of mice where the brothers and sisters all in the same litter. Some have a full corpus callosum, some have partial agenesis, and some have complete agenesis. So we now then had a way to study mice with partial agenesis. And so Elliot and I supervised this very brilliant medical student who completed his PhD with us. And he's now a psychiatrist in Australia. And Tim did MRIs on these mice. And maybe you can see these are the partial agenesis mice. You can see a little bit of crossing here. And this is the complete agenesis mice. And the reason I'm showing you this is because you can do some fancy analysis on mice. And this has also been done on humans as well in Elliot's group, where you take the brain imaging data that you have and you parcellate it into the different regions of the brain. And then you can draw a line, if you like, between different areas of the brain that are actually connected. So this is just a graphical schematic of that. And the dots here are the different areas of the brain. And the size of the dot tells you how much that area is connected with other areas of the brain. So a large dot has a lot of connections going to, it's like a node of connections. And so what Tim did was look at the control mice with full corpus callosum from the same litter. He looked at mice with complete ACC. He looked at the partials, and he did something which is called a virtual callosotomy. So what we wanted to do here, and this had been done in Elliott's lab in people as well with corpus callosum disorders, is we wanted to see which areas of the brain are connected in the mice with complete ACC. And you can see it's really, just by looking at the pattern of the dots and the lines between them, you can see it's really quite different to the controls. And it's really quite different to the partial agenesis mice. And what this study really found or reinforced is that if we, what we can do is to take the control mice and now just take the data and remove the callosal connections from the data and then look at what's left. So this is what we call a virtual callosotomy. And the reason we wanted to do that was to understand, is it just that you've removed the corpus callosum and everything else stays the same? Or when you remove the corpus callosum during development in corpus callosum disorders, is there a reorganization of the remaining fiber tracts that are there? And so does that make sense? Okay, so if you compare this with this, you can see it looks really different. So, and this one actually looks more like the controls. So this tells us that there are plastic mechanisms during the development of these fiber tracts that actually change in people and mice with corpus callosum disorders. It's not just that you take the corpus callosum out, it's that you get reorganization of the remaining tracts as well. Make sense? You look a bit confused. I'm worried. All right. We could talk more about it a bit later if you like, and I can run through it a bit slower. But the main thing I'm trying to get across is that corpus callosum disorders are highly variable between people from a structural point of view. From a cognitive point of view, there's much less variability. So this tells us that you can get similar cognitive outcome with differently wired brains. That's how amazing the brain is, right? So let's move on. Okay, so that's all I was going to say about how variable corpus callosum disorders are. I just wanted to talk about the genetic diagnosis now. So why identify the genetic cause of a brain condition? Because this is the first and a very key step required for something that is coming and is called personalized medicine. So treating individuals with clinical treatments, perhaps based on their genetic mutation. And so we can tailor treatments and therapies, hopefully in the future, on that to each individual. It also often provides an answer for the family and in terms of how did this happen? What caused it? And that can be very comforting to know. It can provide prognostic information potentially. And this is where we really want to link it up with the kind of longitudinal studies that you've heard going on in Lynn Paul's group and Warren Brown's group and other groups around the world too, looking at longitudinal assessments of people over time. And then we want to link that to the genetic condition that they have. So then new people who have the same diagnosis will be able to give better prognostic information too. And this also provides, having a genetic diagnosis provides an ability to provide genetic counselling around family planning and also potential to develop targeted treatments. Oops. Just go back. All right. So the genetics, why is it so powerful? Really, it started with the completion of the Human Genome Project. Many of you might have heard of the Human Genome Project. It started in 1990. It was heavily funded by the NIH, but also other groups around the world. And there were different sites where groups were sequencing the human genome. And the project was completed in 2003. And this is what we call, you know, just a major project that was going on around the world. It cost billions of dollars to finish that, $3 billion to read 3 billion letters in the human genome, and took 15 years. So that was in what, 20 years ago. It now costs $1,000 and we can sequence a genome overnight. It's incredible how much genetic technology has advanced in that time. And it's been just an amazing transformation of healthcare across all different clinical conditions. Okay, so DNA is what we're talking about when we're talking about genetics. It is this double helix instructive code that's found in the nucleus of every cell in the body. And it's a code made up of 4 different letters, A, C, G, and T. And every human cell has this. It's organized into 23 strips called chromosomes. And the order of the letters determines the information, for building. I'll just try and go back here. Yeah. For building a person. It's pretty amazing. So in the DNA, I think this is really important with regards to the genetic diagnosis. There are 25,000 genes approximately in the human genome. And each gene provides the instructions for one protein. Not every gene is expressed in every cell. That is, when we say expressed, makes a protein in every cell. And there are huge stretches of regulatory areas of the genome that dictate which genes are expressed in which cells when. Okay, so every cell contains 2 copies of the gene. Okay, so health conditions can be caused by genetic or non-genetic means, and there are some non-genetic causes of corpus callosum disorders. We actually don't understand them at all very well, but some maternal infections and some fetal alcohol spectrum disorder is associated with corpus callosum abnormalities. But for the most part, They are genetic conditions. So, okay. An example of a non-genetic health condition is COVID, like an infection. An example of a genetic condition is like cystic fibrosis, which is alteration of genes in this protein called CFTR, and it causes respiratory problems. Over 6,000 genetic conditions have been identified across different organ systems. So there are three main classes of genetic conditions. The first is called a de novo mechanism. And this is when neither parents are carriers, and the genetic mutation just happens by chance in the affected individual. So in this case, there's very small chance that multiple children would be affected. And you can see in 3 billion letters, many of us have changes in our genome and they're not pathogenic. They don't cause a condition, but occasionally they do. And this is just the luck of the draw. 2 other mechanisms are autosomal recessive mechanism. This is where one or both parents are carriers. They have a genetic mutation, but it actually doesn't cause any symptoms. And when they have children here, you can see that the children may be affected. So the blue is the affected individual who got the affected allele. So remember, these are chromosomes here. And each person has 2 copies of the gene, except for X-linked, except for the sex chromosomes. Okay. So here, this child got both affected copies, and so they're affected, but these 2 children are carriers, and this person has none, is not a carrier and doesn't have the affected allele. The next type of condition is an autosomal dominant mechanism. This is where, in this case, the dad is the carrier. So you can see the mum has unaffected alleles. They're in kind of brown here, and the blue is the affected. He doesn't have the condition necessarily, but well, he will in a dominant, he will have the condition. He's affected, and he can pass on either the affected or the unaffected allele when he has children. So in this case, 50% of the kids will be affected and 50% won't be affected because the mother isn't a carrier. Okay. There is one more type of change, which is quite complicated, but I'll try and go through it. This is where the mutation so those mutations I was talking about happened in sperm or the egg stage. So every cell in the body is affected. There can be mutations. So when a baby is being formed, it starts out from the egg and the sperm coming together. The egg becomes fertilized and then starts to proliferate, generating more and more and more cells. It has to generate all the cells to make a baby. And as those replications are occurring, mutations can be introduced into the genome at that point. That is called a somatic mutation because it's not in the germ cells. And a somatic mutation can occur in specific organs because when it might have occurred when that organ was being formed. So in those cases, it's a kind of variation of a de novo mutation because it's only happening in that one individual, and it's actually only happening in one or more areas of the body that happen to be being formed when that mutation occurred. So some cells are affected, others normal. The blood could be unaffected, but you can find the mutation looking in skin fibroblasts. I'll talk a bit more later, maybe in the question and answer, about skin fibroblasts and why and how we make models, cellular models from them. It could be a chromosomal abnormality. That would be where there's a deletion of part of the chromosome, or it could be what we call a single nucleotide change, a polymorphism, which is just a change in one of the base pairs. And it has been reported in many cases of brain disorders. So you could have a person with just one part of their brain affected, one side, one hemisphere even. Okay. All right. So how do we do genetic testing? Most people probably have had their affected individual might have had a microarray. This tests for loss of gain of large segments of DNA. We call them copy number variants. So where they're looking at chromosomal changes, really broad changes. Okay. The next kind of test would be an exome sequence. So this is currently where most clinical tests would be. So an exome is the areas of the genome where the genes lay. So remember I said there are about 25,000 genes in the genome. There is a lot of the genome that doesn't encode a gene. We think that most of the rest of the genome is encoding we call them regulatory elements, the parts of the genome that regulate when, where, and at the exact timing that gene will be turned on to produce a protein. So we don't, sequencing the 3 billion letters of the genome, we may be able to do it quickly now, but analyzing those 3 billion base pairs, we are still developing the technology to be able to do that. So we don't tend to do whole genome sequencing yet, but it's something that we will be doing a lot more of, and probably even in the clinical setting at some point. It's actually the technology to analyze the sequence that is holding us back at the moment in the genetics field. But analyzing the exomes is kind of state of the art now. So most people will have had exome sequencing. And the reason I wanted to clarify this is because for the 50% of people that we haven't been able to provide a genetic diagnosis for yet, it's possible that, of course, we've done exome sequencing and we didn't find anything. But it's likely then that there may be some mutation in the regulatory elements that we haven't analyzed yet, potentially. So let me just, okay. So genetic conditions are, here we go. Genetic conditions are often caused by just a single letter change in that 3 billion letter genetic code. So it's actually not easy to find them. It sounds easy, but it's not easy because all of us have changes in our genetic code, all of us, but most of them don't cause disease. So what we have to do is the kind of gold standard way, if somebody has had a genetic variant identified, what we will do is immediately look in the literature to see if anybody else has identified a mutation in that gene that might be causing the same outcome. We call it a phenotype, the outcome. And we also go to other websites where other researchers upload their genetic mutations from the families they're working with to see if they have any, because what we want to see is the same genetic mutation in another person with the same phenotype. If we see that, then we can be more confident that that mutation is the disease-causing mutation. In disease, in this case, it's a broad term, but you could say corpus callosum condition. So, my goodness. Let me just go back here. So that's sort of how we identify whether the gene might be disease-causing or not. But if that mutation hasn't been seen before, if it's not in these matching databases, then we call it a variant of unknown significance. And so that means it needs research to figure out whether that variant might actually be disease-causing or not. So this is equivalent to finding a single typo in 1,000 copies of War and Peace. So reading through War and Peace, 1,000 copies, and finding a single typo in that. Or 300 copies of the entire Harry Potter series. So just to give you the perspective of how complicated it is. Every individual listening, has about 3 million letters that differ between each of us. 3 million letters that differ. So it's complicated. So this is a study showing some commonly disrupted genes in disorders of the corpus callosum. And what we're showing you on the x-axis here is the chromosome. As I mentioned before, we've got 23 chromosomes. And so this is called a Manhattan plot because it looks like, skyscrapers. And this little red line here is kind of the significance of that plot and how many people might have had the disorder. A number of things sort of go into this, what's on the y-axis here, which is the likelihood that it might be disease-causing. And so if it rises above that, then, these are some of the very likely genes that might be involved in corpus callosum disorders. At the moment, there are literally hundreds that have been identified that can cause a corpus callosum disorder. So it's very variable. The genetics can be extremely variable. And that's what makes it kind of difficult to study sometimes, because if it was like cystic fibrosis and there was a single gene disorder, then all your efforts can be poured into that gene. But here we're talking about hundreds of genes that can cause a corpus callosum disorder. So, and then you've got the huge variability between people in terms of the structural phenotype as well. So it's complicated, and you may be frustrated and wondering, why isn't the research going faster and answering all these questions? It's really because it is a very complicated group of conditions. And so, so I was going to move into the last part of the talk now, which is why are so many genes involved in corpus callosum development? Because forming a corpus callosum is also very complex. But maybe we want to have some questions at this point about the genetics, and then I can answer any questions you have about how the genetics is done, or was it all clear? Yeah.
being able to tailor treatment and therapies. Is that some way that prevented there may be treatment?
Right. So I talked about this in the research session a little bit. So when people get a genetic diagnosis now, usually what they do is go straight to Google and start looking for other people that might have, to learn about the genetic disorder and to look for other people that might have the genetic disorder. And so actually a bunch of family organizations are now cropping up around specific genetic conditions. And a lot of those, so I gave some examples of about 15 different family organizations that are associated with single gene disorders that have corpus callosum disorders, amongst other things. And each of them are trying to attract attention and research to those genetic conditions. And they're readying themselves for clinical trials around those specific genes. So yes. Now, when you say, could there be a treatment? There's going to be treatments for various aspects of the symptoms. And so some people have epilepsy, some people have cerebral palsy, some people have intellectual disability. And so it depends on what the gene actually does. That's where knowing what the gene does is super important. And then developing ways of screening for drugs. So there's kind of two ways that family organizations are kind of attacking this. One is we don't care what the gene does. We just want to screen small molecule libraries, they're called, that you can get, or drugs. So there's huge panels of hundreds and hundreds and hundreds of FDA approved drugs. And if there's a way they can screen which ones might be effective, then they might want to do a clinical trial in that. So to do that, generally what they do is develop cell lines from the people that have that genetic condition. And we can do that literally by taking fibroblasts from the skin and transforming those, growing them in the lab and transforming those fibroblasts into neurons. And then what we, so those, if you take the skin from a person with a genetic condition, those neurons will have that genetic condition. And then you can compare them to control neurons. And if there's some feature of the cell that's different, maybe the length of the axon or the way the neuron fires, then what we would do is screen drugs against that and try and rescue that. And then we would know that we have a drug that might be able to be used in that group. But then they have to run a clinical trial and it might fail. We don't know if it would then rescue the phenotype in the person. But that's like in very broad terms how these kind of personalized medicine things are coming about. So that's if you don't care what the gene does, we're just going to try repurposing every drug. And some of the family organizations are doing that or supporting research to do that. The other way is trying to understand what the gene actually does. And one of the best ways to do that is to make the mutation in a mouse. And then you can study that mouse in detail and try and figure out how it affects whatever it is you're trying to understand. Yeah.
Apologize if I didn't catch it in the presentation. Is my son's condition like DCC genetic to the degree that I should have my other kids tested to see if they are carriers, or is it mostly that spontaneous?
Yeah, so that all depends on the inheritance. So I did talk a little bit about whether it's de novo, meaning it only happened, that mutation only happened in the affected individual, or whether it's autosomal recessive or dominant mutation. So it all depends on the mutation. And firstly, hopefully you have a genetic diagnosis, because 50% of people don't yet. And then we would be able to tell you, or at least a genetic counsellor would be able to help you with regards to having your other children tested, or whether they might be carriers.
Genetic tests they can run for the other kids.
Oh, yeah. Certainly, like if we know the gene, we don't even have to sequence. We can just look for that gene, the mutation in that gene. That's really easy, really easy.
First, that's the child to determine if it was an inherited gene.
Yeah.
So,
Yeah. This is why genetics can be so powerful for families too, and why we are really desperate to identify the genes in every condition that we can, because it's very powerful knowledge to have. Right. Elliot. The families are asking where is the best place to get a genetic test? Well, any kind of genetic, if they haven't got a gene for their child. So generally, this is done by a clinical geneticist. And certainly at WashU, we have a whole department of clinical genetics. And Elliot has a clinical geneticist that he works with as well. Or you can enroll in a research study that will probably take a lot longer, but we could also look for the gene. If you go to a clinical geneticist, they will only do a microarray and a whole exome, because they definitely won't have the expertise to do whole genome sequencing. Yeah, we, as I mentioned, pretty soon we will be doing whole genome sequencing, and it will be much easier using literally machine learning algorithms, no doubt about it. Yeah. Yeah.
So that's interesting, because they want to do this again at some point. Did I misunderstand you? When I heard you say there's still a possibility that there is some genetic link there.
Yeah.
Have that been located yet?
Yeah. As I said, I mean, you can correct me here, Elliot, but most corpus callosum disorders are genetic. That's what, of course, we're the genetics people, but most are genetic. Unless there's fetal alcohol spectrum disorder, or, drug addiction, other kinds of addictions that were, the mother had, that will affect the baby. But if that's not apparent, it's most likely to be genetic. So the fact that you don't have a genetic diagnosis doesn't mean it's not genetic. It means that the technology isn't there, or it's, a lot more digging needs to be done. Yeah. And so it is the case that the research studies have, although this is changing, like it's being pushed out into the clinics more, but the research studies are more intensive in trying to identify those, the genetic mutations. And so, the researchers are the ones that are pushing the boundaries in the genome technology, right? And the analysis of the genome. So, yeah. But I can tell you that the regulatory elements, the work being done on the regulatory elements that regulate where and when the gene is expressed, that is, very new, but it's extremely powerful, right? And these epigenetic mechanisms, that they're related to that. So even the chromosomes, they're coiled up inside the nucleus, and they have proteins that sit down and open the chromatin and allow other proteins to bind there and then express the gene. So there's a whole way that the DNA is wrapped up inside the nucleus, because you don't want all those 25,000 proteins expressed in every cell. That's what makes brain different to heart, different to lungs, different to muscle. The different cell types are because of the combination of genes that are expressed in each cell type. It's very complicated. Yeah. 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 Well, we have previously done all of our genetics with Elliot. So if you come to WashU and we took a saliva sample, we would send it to Elliot. But if you want a clinical, like if you want it, and Elliot is going to batch them up and sequence them a year from now or whenever he's doing a sequencing batch. If you want a faster diagnosis, clinical genetics, like where your health insurance might pay for it even. And so you can sign up with you could get a referral to see the clinical genetics group. And that's what I would recommend first if you haven't had any genetic testing, because they'll run a microarray like quickly. They'll do exome sequencing quickly. And that's where you're most likely to find, the genetic cause in 50% of the people. But yeah. Right. So that's maybe another really good point. The technology, as you just heard, has just been rapidly gaining, increasing. And so if you haven't had genetic testing done even, 10 years ago, it's definitely worth and you had no genetic diagnosis then, it's definitely worth getting genetic testing. In 5 years ago, Elliot, do you think, or 10 years ago? Yeah. So like if you had a genetic test done, like microarray or something done 10 years ago and they didn't identify anything, then you definitely want to be, I would definitely want to be tested 5 years ago. That's how rapid it is, right? So if you haven't got a genetic diagnosis, keep trying is what I... Oh, there is. Yeah, there definitely is. Yeah, because I'm not a doctor. Elliot is a doctor. So what is the route by which people get genetic testing done in the States? Clinical geneticist. Yeah. Maybe it differs by state here as well, which I'm not sure about. Yeah, but certainly, I mean, I only know about WashU, and they're the clinical genetics group. Yeah, you can get genetic testing through there. Just I don't know if you get a referral there. Yeah. Yep. But yeah. Oh, right.
Your dollars.
What did you say, Elliot? In your healthcare? Yeah. Okay. He's gaming the healthcare.
Yeah.
What he said was if you haven't used up your genetic testing dollars and is going to roll over into your plan for the next year, then get it done. But I think if you have a referral, then you can get it done through your healthcare provider. Yep.
The diffusion scan, how available is a scan like that?
Yeah, only in research. The diffusion scans. Yeah. Because they're really for research. At the moment, we are trying to correlate the connectivity with the cognitive outcome, but we haven't got enough data to be able to say, oh, that's what we'd really like to do. However, what's sort of becoming more, and I haven't really talked about this with Elliot, so let me just go for it. What we're really finding is that you can have very similar cognitive outcomes from differently wired brains. So I'm actually wondering how useful it is to know the exact wiring. And I'm sure there'll be some degree where it's useful and then some degree where it's not useful. You know what I mean? Like if you've got no connectivity down the back and all of it's up the front, then that's probably useful. But if you've got a smattering all over, but it's less, I'm not sure how useful that is, right? So we do other kinds of scans. They're called functional MRI scans. And that's where we're looking at which areas of the brain are active during either resting in the scanner, just lying there, and your brain is active. Another thing that we do is actually ask the participants to do something while they're in the scanner, and then we can compare which areas of the brain are active using blood flow. Yeah. Right. Shall I talk a bit about the different genes and how they actually affect corpus callosum development now? Yep.
White and are both high functioning, are both isolated, complete.
Oh, yeah. Yeah.
I guess how common is that if you have parent and sibling that both have the same disorder, same version of the species?
Yeah. Well, certainly Elliot's leaving. No, he's getting some water. We've got a number of families like that. Yeah. Even a grandparent, generations, multi-generational. Yeah. Yeah. Do you know the gene? Okay. And actually it is the complete ACCs that we are the ones where we don't know the gene the most, if you like. We know the gene for some of them, but it's the high functioning completes that have clinically, I'm super interested in them, but clinically they have been on the back burner, you could say, because you can imagine there's a huge number of genes where the kids are really severely affected and they've got all these other organ system problems as well. They've been the ones of the focus of, the pediatric neurologists. When people are doing pretty well, they don't tend to go to the neurologist, right? So then they don't get the genetic testing then. So we know far fewer about the genes in the complete group. Yeah. There's no ridiculous questions.
Idea artificial.
Yeah. Okay, so here's where it's really important to know what is the corpus callosum. It's not just the bit that we show on those images of the bit that's crossing the midline. It's, maybe I could show some of the images I'm about to show about how the genes affect the corpus callosum, because I was going to talk about corpus callosum development and actually the main cause of complete ACC, which we do know from structure. Could I do that? And then we can come back. All right. So at least 400 genes are involved in forming a corpus callosum. What I've done here. Oh, there it is. Put down the pointer. All right. So both Elliot and I work on early brain development. So before birth, the corpus callosum forms between 12 and 20 weeks of gestation. And we're interested in those brain developmental events that set up the wiring of the brain and how that affects cognitive function. All right. So as I said, identifying the gene is step one, but what do? How can we study genes in corpus callosum development? Here's an adult human corpus callosum. This is a mouse, and this is what a mouse corpus callosum looks like when you cut the brain this way. So mice have a smooth brain. They don't have the bumps, the sulci and gyri. And what is amazing now is that really we can study every aspect of the corpus callosum in a mouse. We can trace the axons. We can manipulate the genes. We can label them in just one hemisphere. We can take the gene out in specific parts of the brain. So we've got a lot of tools at our disposal. So that even though they're mice, they're very powerful in understanding what the gene does. And so here, this is really tiny. Here is a control mouse that has a green corpus callosum here. And this is a sagittal view, but you can't see it. And here is a mouse with complete ACC, and he has little probes bundles. This is an MRI image, so we can also use MRI in mice as well. And we can label the cells that give rise to the corpus callosum in one hemisphere only. And when we do that, actually what you see, so the cell bodies are sitting up here. So this is the neuron. And it's sitting, there are 6 layers in the cortex of mice and humans. And the corpus callosum neurons sit in layer 2 and 3 and layer 5 for the most part. They then send their long wire out. We call that an axon. And this is the axon. So the cell bodies sit up here and they're actually sending their wire down here. They're forming branches in that layer, and that's why it looks so dense. And then this is the white matter tract. This is the midline here, one hemisphere, the other hemisphere. Here are the axons crossing over the midline and innervating the other hemisphere. So innervating means forming a connection with the neurons over here in this hemisphere. And you can see some of them are connecting here and some of them are connecting there. So, so that's what a mouse corpus callosum looks like. And we know that there is another cell type in the brain which are called glia. So the neurons in the brain are the ones that encode the information, and they are electrically excitable cells. So they have electric currents that go throughout the circuit. You can just really think of it like an electric circuit, your whole brain. And there are neurons firing all over the brain at the same and different times. But there are other cell types in the brain, and these, one of these are glia. So glia comes from an ancient word that calls they thought originally these cells were like glue that helped to stick everything together. But it turns out that glia are much, much more important than just glue to bind everything together. And in the early developing brain, glia are really important for forming fiber tracks. So in the mouse, here we're now focusing on, remember, this midline area, which is where, you know, the fibers all come together to cross the midline. And you can see I've drawn a picture here of the mouse corpus callosum in red, but in green are the glia that are surrounding this whole area where the fibers are going to cross the midline. And actually, these glia, they secrete molecules that help to guide the axons across the midline. So remember, I said that the brain is super plastic during development. These axons are not predetermined, like exactly where they're going to go. They actually look for guideposts as they're growing to form the connections. Okay. Now, I put this up because the genes that go into forming a mouse corpus callosum are highly conserved. And that means that they're also all expressed in people as well. And in fact, they're not just expressed in people, they're evolutionarily conserved even in flies and worms. Now, flies don't have a corpus callosum, but they do have a nervous system with axons that cross the midline, and the same molecules are expressed by glial cells in those animals as well. So when something's evolutionarily conserved, it's important because otherwise evolution would have gotten rid of it. But when it's found in, you know, everything from a worm or a fly or a fish all the way up to people, it's a mechanism that was reused and reused during evolution. So that means we can use animals to study these disorders. All right. So I wanted to talk a little bit about what is the cause of complete agenesis of the corpus callosum. And the key here, so now we're not talking about the genes specifically, we're talking about how the brain forms. So here's a person with a neurotypical, there's the corpus callosum, and when you, so that's slicing the brain this way, and the bottom picture is the same person slicing the brain this way. So you can see the corpus callosum here crossing from one side to the other. In this person with complete agenesis of the corpus callosum, what you notice, again, here are the probe spundles, so the same, we're slicing the brain this way. But what you can really see is that this little piece of tissue here is called the septum pellucidum. And in the neurotypical individuals, that septum pellucidum is fused together. So you can see that there. In people with complete ACC, the septum can still be there, but open. So you can see there's a gap here, and you see that tiny little piece of tissue here and here, they're open like that. So during development, something didn't happen. So let me tell you what that is. So firstly, to convince you that this is important, we did this study with Elliot's group, and we took scans from Elliot's cohort of people that he's been working with. And we looked to see how many people with complete agenesis had these open septum. And 100% of the cases have open septum. So it's again a very highly conserved kind of structural change, like colpocephaly and probe spundles, actually. So how does this happen? There's a bit of a delay on the slides. Okay, so in order to explain that, I need to explain early brain development. So as the nervous system is forming, it forms a tube first, and down the front end of the tube, it forms a vesicle that will form the cortex and the front part of the brain, and then the hindbrain and a structure called the cerebellum, you might have heard. These form behind that. And this starts out as a tube, and then there's some bulges that start to happen as the cells continue to divide to form all the cells in the brain. And so you have, and initially, this is a mouse brain, this is a human brain, and this is a human brain. So initially you can see that it's kind of one vesicle that forms. And then to get the two hemispheres, what happens is that the tissue starts to invaginate and form a cleft between the two hemispheres. So it's connected at the bottom. I can't do this and hold the thing at the same time. It's connected at the bottom, but there's a gap, right? And that gap is called the interhemispheric fissure. And okay, so you can see this is a 12-week gestation, an image from a 12-week gestation fetal brain. And you can see the interhemispheric fissure here. This is looking front on, anterior view, a posterior view, and looking down on top of the brain here. And this is the mouse brain throughout. This is all at 12 weeks gestation. This is the mouse brain early on. And then as it develops, you can see it also forms this interhemispheric fissure. Now, the corpus callosum is actually going to form, so what you're looking at in this image is the mouse brain front on, like that. And so you're looking at the front of the brain there. And the corpus callosum fibers are going to cross at about this level. But imagine there's a gap there. So, well, not imagine, there is a gap there. You can see the gap there, and the fibers don't cross over a gap. So how are they going to cross? Well, they cross because there's a specialized population of glial cells that arise. They're born in the middle of the brain, and they migrate forward on either side of the fissure. And then they, once they get into place, they do something which is called differentiate. So they start out as a bipolar cell, like a very skinny kind of cell. And then they transform, they differentiate because other genes are expressed that control their differentiation. And they become these multipolar astrocytes, they're called, but they're just a mature glial cell that forms a bridge. And that fuses that interhemispheric fissure together and forms the bridge, and the callosal axons grow across that bridge. So if that bridge doesn't form, the axons can't get across, and instead they grow front to back and they form Probst bundles. So here we know that this also occurs in people. This is a developing human brain at either, what is that, 13, 14, or 15 weeks gestation. So 3 different brains here. And I have stained these sections. So these are sections this way, coronal sections. I've stained them for the glia. So these black sort of cells here are the glia. Now, these specialized glia are called midline zipper glia because they literally zipper up the midline. And what you can see is at 13 weeks gestation here, the 2 sides of the brain, the midline, they're separated, but the glia are getting into place. And then at 14 weeks gestation, you can see it's starting to fuse here. And this is the septum on either side. This is the septum pellucidum here. And then you can see by 15 weeks, you've got quite a lot of corpus callosum fibers crossing over this point. So that is how the corpus callosum gets across the midline. And in a mouse, you can see here the same thing happens. So here the glia are in place. The glia are in brown here, and the zipper is open. And here the glia have formed these multipolar astrocytes that are knitting together that midline, and the zipper is closed. And I have a scientist in my group who's sitting down the back here, who is currently figuring out exactly how they fuse. We don't actually know the molecular genetic basis for how they fuse, but we're figuring that out. And this is an MRI of a developing human fetal brain. And I'm showing you these only to let you know that this is not just something that occurs in mice. The same thing occurs in human brain as well. And so we have to study human brain to show that the same thing occurs in human brain that occurs in mouse brain. So this is a 13-week gestation human fetal brain. And this is the septum here. And you can see maybe this is an MRI scan now. And you can see that even though it's the same brain, some parts have fused, like here, and some parts haven't fused yet, but are undergoing fusion at this 13-week gestational age. And this is what it looks like from a different angle. So this way, now we're cutting the brain in a horizontal way in the mouse, and we're looking down upon this area that's fusing. And we're staining it with different stains. The purple one stains the glia, and the green one stains the fissure. So you can see, and this is mouse at embryonic day 14. So mice have a 20 days of gestation and then they're born. And the corpus callosum in the mouse forms between E14 and E17, embryonic day 17. So here is E14 to E17, and we're just looking at the fissure closing up and these cells migrating forward. And then you can see this interhemispheric fissure is getting smaller and the area where the glia are is getting bigger. And that's providing the bridge where the corpus callosum axons can cross. And we know that the corpus callosum axons cross only in this area because in white here, we have labeled the corpus callosum, the first axons that cross the corpus callosum. And we know that they are tracking down along the interhemispheric fissure. And here, this is a higher power view of that area there. The green is the fissure and the white are the axons. And they're just tracking on. They're not crossing where the fissure hasn't fused, and they grow down to the area where the fissure has fused, and they only cross there. So I think that's about it. So this is just a schematic. Different genes are involved in forming this corpus callosum bridge, if you like. And this is the normal case where the corpus callosum axons approach the midline. These are these special glial cells there, form the bridge, and the axons cross over the midline. Where they are not able to cross, in this case, it's a gene called nuclear factor 1, then they form these Probst bundles. Does anybody have any questions about that? Yeah. Right. So so different genes affect the development of those glia. And I could actually show you, if we go back to the slides, I'll show you a gene, some of the genes on this slide. Okay. I'll just skip over this. Seems to be going backwards. We'll keep going here. Keep going. Could you just click through a few slides and then I'll tell you when to stop? Keep going. Okay, so just go back one. So this is a family of genes called nuclear factor 1. And these genes were identified some years ago. And here is a paper from 2007 showing 5 patients that have mutations in a nuclear factor 1 gene. Some kids have thin corpus callosum, some have complete agenesis, some have partial. Next, well, I'll do the slides. And I'm just showing this because nuclear factor 1 regulates the development of those glia. So in those kids, there are 3 different genes in this family. They're on chromosome 1. This one's on chromosome 9. This one causes something called Soto syndrome, NF1X, A and B. But across all these 3 syndromes, the kids have enlarged brains, enlarged ventricles, corpus callosum disorder, and developmental delay or intellectual disability. And so that's the enlarged ventricles. You can see they also have colpocephaly. And I want to get to the point where they have some dysmorphic facial features as well. Here are some of the mutations. I'm going to keep going. These are mouse. I had a lot of slides here. These are different mouse brains, different mouse knockouts. Okay. So, so they regulate the differentiation of those glia from the bipolar cells, a line along each side of the midline, to the point where they actually become differentiated astrocytes. They regulate that one little step. And so that definitely is what is causing the corpus callosum disorder in those kids. But we're also looking at how come they have enlarged brains as well. And those genes do some slightly different things in different cells, but we're looking at that. This gene is now becoming the most commonly affected gene, 5 minutes. Yep. In corpus callosum disorders, it's called deleted in colorectal cancer. It was first identified as a colorectal cancer gene, but it's actually not that well associated with colorectal cancer, but it is one of the most common genes that's in corpus callosum disorders. And the people that are affected commonly have something called mirror movements, congenital mirror movement disorder. And that is where they are unable to inhibit one hand from moving when they're moving. Like if you ask somebody to do that, they're unable to inhibit the other hand from doing that. So it's a symptom that it's quite obvious to see, and they know it themselves that they have it. So this gene is, it sits on the membrane of the axons. So remember I was talking about the axons growing across the midline. So at the end of the axon, there's something called a growth cone, and literally it's like this. It has finger-like projections, and it's sniffing around for guidance molecules that are expressed by the glial cells that help direct where the axon is actually growing. And these are the receptors for those molecules in the environment. So that's what I've drawn here, DCC sits across the membrane. This is the membrane of the cell, and it binds to different molecules in the environment. And it regulates the differentiation of the midline zipper glia. So when the zipper glia, oh dear, I'll just skip to this. It regulates the formation of these midline zipper glia that I was talking about before. And we know, nuclear factor 1 genes regulate it. We also know molecules in the fibroblast growth factor family also regulate it. But I think I'll stop there because we're out of time, and maybe other people have some more questions. These are some of the people that we're working with and some of the people who are outside. Are there any other questions? Oh, yeah, sorry. Would you say that again? RAC1. Yeah, RAC1. Yeah. Oh, okay. Okay. Well, RAC1 is a downstream signaling molecule of DCC. So maybe I could talk to you about it at the end of the session. Yeah, for sure. Anybody else have a question? Thank you very much for your patience. I know that was can be confusing and complicated.